If you're a Western-trained manual therapist or practitioner, when you compress tense, tight, soft tissues using thumb pressure, for example, you're probably aware that a number of predictable and easily explained effects are taking place. Digital pressure obviously produces mechanical stretching of the connective tissue. It also stimulates mechanoreceptors, so influencing pain perception, and of course it will temporarily produce increased ischemia, so that on release of the pressure, a flushing of fresh, oxygenated blood occurs. Pressure also triggers the release of endorphins and enkephalins. So we can see that a simple biomechanical action has obvious mechanical stretching effects alongside neurological, circulatory and endocrine ones. Now, the very same pressure applied by someone trained in the Eastern traditions of Ayurveda or traditional Chinese medicine might be thought of as altering the flow of prana or qi, with an energy-related outcome on health. In cranial work, it's also possible to see that performing the same activity, say for example fourth ventricular compression, might be having predictable biomechanical influences by causing a degree of slack in the reciprocal tension membranes that attach to the inner aspect of the skull, potentially influencing venous sinus drainage. Or if your understanding of the cranial approach is biodynamic rather than biomechanical, this same cranial hold might be conceived to be having a transmutational, fluid-driven effect on the healing processes of the body. Some remarkable images to be found in the chapter of the book by McPartland and Skinner illustrate some basic concepts of biodynamic cranial theory. These suggest that the embryo is a biodynamic archetype, serving as a blueprint for the body's ability to heal itself, so that the formative, resorbative and regenerative fluid forces that organize embryological development may also be present throughout life. Biodynamic cranial practitioners postulate that the external forces generate a spatial orientation in the embryo. This becomes expressed in the material plane by fluid forces, perhaps by electromagnetic water-hydrogen bonds, generating a matrix that governs the embryo's development. The tensile fluid forces required for this process were demonstrated by Schwenk in 1996, who, using micropipettes, injected streams of fluids into water. Boundary surfaces arising between the moving fluid and the still water vortexed into organic forms. By changing the fluid density, or the injection speed, different forms were created. In some experiments, the tensile quality of the fluid matrix produced shapes that closely resemble the migratory path of the neural crest cells in the embryo. In other experiments, the spatial orientations of fluid in a fluid suggested central nervous system formations in the embryo, complete with dura and pia, cerebral hemispheres, and a corpus callosum connected to the hemispheres. These concepts are more fully explained and explored in the book. Biodynamic cranial practitioners correlate these concepts with Sutherland's description of the tide, acting as a fluid within a fluid, expressing a tensile quality with the ability to direct force. More recently, the reaction of genes to hydrostatic pressure during embryogenesis has been termed the morphogenetic mechanism. Van der Waal likened genes to the clay that forms a piece of pottery. Clay can't shape itself. It requires the hands of the artist, and the hands of the artist can't act without the mind of the artist. From a biodynamic perspective, clay represents the genes, the hands represent the fluid forces, and the artist's, therapist's mind represents the expression of those forces. The therapist practitioner working with the biodynamic model will be attempting to influence these fluid forces, possibly by means of a process of entrainment, as discussed in the text in chapters 2 and 4. Biomechanical cranial approaches regard the structures and functions of the cranial mechanism in quite different ways. Focus would be more toward evaluating structural relationships and their functional influences, including venous and lymphatic drainage, rhythmic cranial pulsations, and dysfunction involving sutural articulations and fascial status, as well as the muscular attachments to the cranium and the relationship between the cranium and the cervical spine. In recent years, both schools of cranial therapy have shown particular interest in autonomic function, as represented by the Traub-Herring-Meyer oscillations. These occur between 6 and 10 times per minute and are variously associated with blood pressure, heart rate, cardiac contractility, pulmonary blood flow, cerebral blood flow, movement of the cerebrospinal fluid, and peripheral blood, including venous volume and the body's thermal regulation. Nelson and colleagues relate Traub-Herring-Meyer wave frequencies to palpatory cranial rhythmic impulse findings. In this chart, we can see the specific effects on the Traub-Herring-Meyer oscillations of 15 minutes of cranial treatment. These represent laser Doppler blood flow velocity records before and after cranial treatment in 2 healthy, asymptomatic individuals. The records represent approximately 3 minutes of continuous, unedited recording made within 20 minutes of each other and demonstrate the effect on autonomic balance of the treatment. Individual number 1 is a 55-year-old male. Attention was given to the craniocervical junction for approximately 10 minutes. Individual number 2 is a 25-year-old female. Treatment involved balancing of the craniocervical junction and the cranial base for about 15 minutes. The high-frequency waveform observed in all 4 recordings is the blood velocity variation with cardiac systole and diastole, that is the Traub-Herring-Meyer oscillations. The prominent lower-frequency oscillation, absent in both pre-treatment records, but prominently present in both post-treatment records, is the Traub-Herring wave. It's quite possible that cranial treatment, even when it's focusing on biomechanical structures, may be having a direct influence on these rhythms, and so on both autonomic balance and the subtle fluid dynamics suggested by biodynamic theory. The choice of any treatment approach depends on accurate assessment findings and the interpretations given to these and on the belief system and training of the practitioner. There are probably no right and wrong choices when considering these 2 models of cranial treatment, merely different ways of achieving similar outcomes. And there's no reason why both biodynamic and biomechanical approaches should not both be available to the skilled practitioner or therapist.